Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A Level Biology Unit 5 for May 2021. This is the part 2 video. I already did the part 1 video and I'll put the link below the description box. So let's begin with question 5. It says, the body length of the medicinal leech decreases when touched. This is the medicinal leech. And they say the body length of the leech decreases less each time the leech is touched. What is the name of this effect? This effect is called habituation, and here the answer should be C, because habituation is expressed, as we can see, each time you touch this leech, the body length will decrease less as the stimulation occurs. So that is a learning process. The leech is learning to deal with this stimulation, and the leech is assessing this stimulation to see if it's threatening or not in order to deal with threatening stimulation appropriately in comparison to non-threatening stimulation. So the answer should be C. And for the next question, they say, why is it an advantage for the leech to respond in this way? It is advantageous because the leech can be able to assess threatening stimulation so that it can respond to those more, saving energy by not responding to things that are not threatening. So it filters out what is important and what is not important in order for it to respond appropriately to the important things and not respond to the non-important things. That way it is going to be saving energy that it could have wasted in responding to non-important things, and that energy can be used for something else. Let us continue. Here they say, the photograph shows a fiddler crab, that is the crab they're talking about, and they say in an experiment, the fiddler crab was used to investigate responses to a predator. They say the responses by females and males were compared. A predator approached a group of female crabs from one direction, and the percentage of crabs running home was recorded. This was repeated 20 times. That is important because it means results can be believable. And then the experiment was repeated with a group of male crabs. The graphs show the percentage of crabs running home for both groups of crabs against the total number of times each group of crab encountered a predator. So here we see the vertical axis for both male and female shows the percentage of crabs running home, and the horizontal axis we see the number of predators. Overall, we can see that the females are more habituated in comparison to the males. Comment on the results of this experiment. We have seen from the two graphs that the females are more habituated in comparison to the males, so the evidence shows that males and females respond differently to the predators. Overall, the females are more habituated in comparison to the males when they're exposed to predators. Also, there is variability in the data present after being exposed to 10 predators, as you can see from the graph. So these were what could earn you the 3 marks. The next question is asking which statistical test would be used to analyze the effect of the increase in the number of predator encounters on the percentage of crabs running home. This is asking us to test a correlation. So a correlation is being tested. That means we need to use a correlation coefficient, and therefore A should be the suitable answer. The other answers do not make sense because they do not suit whatever we're trying to look for here. Question 6. In humans, the sinoatrial node, atrioventricular node, and the bundle of His are involved in the regulation and coordination of the cardiac cycle. The diagram shows a human heart. So the human heart is labeled, there is Q, P, and R. This is the sinoatrial node, that is the atrioventricular node. So the first question is asking which one of the following statements is correct. So statement A says Q is the sinoatrial node, that is correct, and R is the atrioventricular node, that is wrong. For part B, it says Q is the sinoatrial node, that is correct, and P is the atrioventricular node, that is correct as well. So the appropriate answer should be a B here. The others are not right. The next question says, the electrical impulse that initiates the cardiac cycle begins without the need for a nerve impulse. What is the term for this process? The process when the cardiac muscle works without external stimulation is called a myogenic process, so the suitable answer should be a B. It's a myogenic process. Let's continue here. A football player undertook regular exercise as part of a training program. During the exercise, there is a change in the duration of the cardiac cycle. They want you to explain why there is a change in the cardiac cycle during this exercise. This change is going to be because the cardiac cycle is going to be shortened during the exercise, and that is because as exercise is carried out, carbon dioxide will accumulate in the blood, leading to lowering in the blood pH. A lower blood pH will be detected by the chemoreceptors found in the blood vessels, and impulses will be sent to the brain. So I say during exercise, a lot of carbon dioxide is produced, and presence of carbon dioxide lowers the pH of the blood. This is going to be detected by the chemical receptors in the lining of that blood vessels, and these will send impulses to the cardiovascular center in the medulla oblongata of the brain. So the responses through the sympathetic system will cause a release of a neurotransmitter, noradrenaline, which stimulates the sinoatrial node to generate more electrical impulses or more electrical stimulation per second, and this will cause a shorter cardiac cycle. So if you answer appropriately like this, you will get 3 marks for that. The next part, it says the table shows the effect of the training program on the cardiac output and resting heart rate of this footballer. So the table shows the results before the training. Before training, we see the cardiac output is 4,500 and the resting heart rate is 72 beats per minute. And after training, we see the cardiac output is still the same, but the heart rate has gone down. The question asks us to calculate the stroke volume of this football player after training. You need to remember the formula: stroke volume is equal to cardiac output divided by the heart rate. So we are just going to feed the values from the table. This is the cardiac output and that is the heart rate. And when we divide, we get 88.24 centimeters cubed. That should be the stroke volume we are required to find. Next, they say after training, the footballer had an ECG. This is an electrocardiogram. This electrocardiogram trace is shown in the diagram. So this is whatever the ECG for the footballer after training. The question asks us to calculate the heart rate of the footballer using the ECG in this diagram. So in order to find the heart rate, we need to find the number of beats, and we need to find the time in which those beats have occurred. If you look at the horizontal axis, we can see here we have 7 seconds. This is time in seconds, so this is 7 seconds. And the number of beats, I have already labeled them. If you count them, they are 18 beats. So here we see there are 18 beats in 7 seconds. So we are going to convert that time to minutes in order to find out how many beats occurred in 1 minute. So here I say the footballer had 18 beats in 7 seconds. And so the heart rate should be 18 beats per 7 seconds. If you divide, you get that. However, in 1 minute, it should be 18 divided by 7 times the 60-second found in 1 minute. And that will give us 154.3 BPM. Or you would say 154.3 beats per minute. The key thing is converting the time from seconds to minutes and then dividing the beats by that specific time in order to find the beats per minute. If you round it off, the answer should be 154 BPM. So here they've given us a second trace. They say 2 hours later, a second ECG was recorded. This is another electrocardiogram of the same footballer, however, a few hours after the original ECG was taken. So let us go to the next page. Here they ask, comment on the changes that have occurred in the activity of the heart as shown in these 2 ECGs. So I want to take you back to the 2 ECGs. Let's look at them side by side and see which changes have occurred. If we look at the first ECG trace, immediately after training, we can see that the heart rate is really, really rapid or fast in comparison to this one here. This one has a slower heart rate and this one here has a higher heart rate. So we can see the first trace has a higher heart rate, the second trace has a lower heart rate. The PQRST for this one here is abnormal in comparison to this one. This one here has a normal or more normal PQRST waves. So we can see that that is another difference between the two. So let's continue here and I'll read the possible answers. So I say the second trace has a more normal PQRST wave compared to the first ECG trace. You can see in the 2 traces. And then the second ECG trace shows fewer heartbeats than the first ECG trace, meaning the second ECG trace shows a slower heart rate in comparison to the first one. And then if you look at the ECG traces, the R wave of the first ECG trace is higher than that in the second ECG trace. And lastly, the first trace does not have a clear S segment, but the second trace has. I put this image here to show you what is a P, because this one is clear in comparison to the two traces we're given. The P is clearly seen here, the Q is seen, the R is seen, the S is seen, as well as the T. So if you can look at this, you can be able to understand what I referred to in this answer here. This brings us to the end of question 6. Let us continue to question 7. Question 7. Animals and plants can respond to light. The photograph shows the front of a human eye. So the first question asks, the size of the pupil changes when moving from dim to very bright light. They want you to explain how these changes occurs. This is easy. They're asking about the pupil reflex. And as you know, if you move from dim light to very bright light, the pupil size is going to constrict. And this change in size is done by antagonistic muscles found within the iris. Remember, they are circular muscles as well as the radial muscles. So here I said, moving from dim to very bright light, the pupil size constricts. Changing the size of the pupil is done by antagonistic muscles of the iris as part of the pupil reflex. The circular muscles will contract as the radial muscles relax, and so that is going to cause the size of the pupil to constrict in order to allow less light to enter the eye, and that is the pupil reflex. Continuing on. The next question says, describe the role of rhodopsin in causing changes in the polarization of the rod cells. Now, rhodopsin is a pigment found within the eye. So rhodopsin absorbs light, and the light splits the rhodopsin into opsin and retinol. This is going to cause the rod cells to be less permeable to sodium ions. The sodium ion channels will close. It means the sodium-potassium pump will keep pumping sodium ions out of the cell, and the inside of the cell is going to become more negative in comparison to the outside. And this is what we call hyperpolarization. This hyperpolarization is going to result into formation of a generator potential, and that generator potential will lead to formation of an action potential in the bipolar neuron, so which will lead to formation of an action potential in the sensory neuron, basically. So if you can look at the underlying parts, the key word here was you acknowledging that rhodopsin will absorb light, you get 1 mark. Another thing is knowing that rhodopsin is going to be split into opsin and retinol when light strikes rhodopsin. And also knowing that when that occurs, the rod cells are going to be less permeable to sodium ions, meaning there will be fewer sodium ions going inside. And that means as the sodium ion channels are going to close, then the sodium-potassium pump is going to keep pumping the sodium ions out of the cell. Therefore, the inside of the cell will be more negative in comparison to the outside, and hyperpolarization will occur as a result of that. Exposure to light controls the germination of seeds and the direction of growth of plants. In an investigation, the seeds of plantains were exposed to light. The seeds were first exposed to red light and then to far red light. The percentage of seeds that germinated when exposed to different periods of red and far red light was recorded. The results are seen in the table below. You need to remember that phytochrome red and phytochrome far red are interchangeable. Phytochrome red is inactive, however, if light shines on it, it's going to be converted into phytochrome far red, which is biologically active. So if you keep that in mind, that can be used to explain the changes that are occurring in here. So let's continue here. They want us to comment on the effect of red and far red light on the germination of these seeds and the processes that occur in their cells. They want us to use the information in the table to support the answers that we have. I began by acknowledging that photoreceptors like phytochromes regulate responses to light in plants, and when they do, they trigger many plant growth and development processes like germination. So germination is one of them, and is going to be triggered by these phytochromes when they absorb light. When photoreceptors receive light, they react to it, meaning in this case, inactive phytochrome red or PR absorbs maximum light and is going to be converted into the biologically or physiologically active phytochrome far red, which is PFR. This one here has a peak in the wavelength about 730 nanometers. So the far red light is sensed by the plants, and if they are not getting enough sunlight, there will be much of it is going to inhibit germination. Metabolic activity and mitosis in the embryos is stimulated by red light and inhibited by far red light sensitive seedlings. So the phytochrome has two identical subunits. There is the chromophore of the phytochrome, which is photoreversible between the two isomers. Of course, I already said this. It's photoreversible between PFR and PR, depending on the wavelength of light. Like I said above here, we have the 730 nanometers as well as the 660 nanometers. The 730, this is for the far red, as well as the 660 being for the phytochrome red. So germination is stimulated by the hormone gibberellin. This is some added information. And you remember these phytochromes are going to cause production of some of the hormones, and the hormones are going to cause growth as well as germination. So this brings us to the end of question 7. Let's continue to question 8. Continuing to question 8, I will begin by showing you the scientific articles. So I am going to scan through the scientific article as you look at it, and then we'll go to the questions. So let's begin with this part here. There is that part. This article is about skeletal muscles and performance as well as aging. So this is the article. I'm going to go through it slowly. I hope you can get time to read through this. It's quite long. So moving through slowly like that. And then to the next page. Like that. There is a lot of information here, inflammation and so on. Question 8. They say, use the information from the scientific document and your own knowledge to answer the following questions. Question A, describe the structure of a skeletal muscle fiber. Refer back to paragraph 2. So like I already showed you the scientific article, I'm not going to refer back, but I'm just going to answer the question, and you can refer back as you look at my answers. They have asked us about a skeletal muscle fiber. Skeletal, very specific. A skeletal muscle fiber should be a single muscle cell comprised of many myofibers. So it's a single muscle cell that has many myofibers and sarcomeres. It is also multinucleated and striated. This striation is because of the repeating bands or repeating patterns of actins as well as myosin filaments. The alternating filaments of actin and myosin are the ones that make a single sarcomere. So that is how I could describe a skeletal muscle fiber. Part B says, explain how myokines may exert endocrine effects on organs such as the liver. Endocrine, this is like hormonal, so meaning it's stimulation. So myokines are released when muscles contract. If I take you back here, they say, this is an excerpt from paragraph 3. It says, furthermore, muscles interact with other organs via the excretion of myokines. These myokines which can exert autocrine, paracrine, and endocrine effects. Myokines support the metabolic function of different tissues such as bones, pancreas, liver, and adipose tissues. So that means they cause the stimulation of some of these organs. So I said, since myokines are released when muscles contract, they are transported through the blood to organs like the liver. Remember, these are myokines, meaning they are released from the muscles. How do they go to the other organs? They have to be transported, and the transport medium is the blood. So they are released in the muscles. When contraction occurs, they are transported through the blood to the specific organs like the liver, and then they can affect the liver by stimulating respiration. So they could stimulate the liver cells in order for respiration to increase, and this could cause increase in fat metabolism, leading to glycogenesis and regulation of homeostasis. I also said this therefore has a direct effect on the muscle itself. So let us continue to part C. Here part C says, explain why the changes that occur to muscle structure and composition in elderly can affect their ability to rise from a chair. They want us to refer back to paragraph 4, and I extracted paragraph 4 and put it here. Let us see. It says, I actually underline the things that I think are important. I say many studies have reported a substantial decrease in muscle fiber size in elderly. They also say that this reduction in muscle fiber size has been shown to be fiber type specific, with 10 to 40% smaller type 2 fibers, meaning the fast twitch muscle fibers decrease, but the slow twitch muscle fibers do not decrease in quantity. Remember, type 2 are the fast twitch muscle fibers. So I said, decrease in muscle fiber mass makes movement more difficult. The decrease in fast twitch muscle fibers, while slow twitch muscle fibers are largely retained, leads to reduction in response to action, like standing, out of a chair due to less muscle innervation. Muscle innervation is about stimulation to action or stimulation to act. The loss in muscle strength makes it hard to perform tasks like moving from a seated position. It is going to take them longer time because that action requires fast-twitch muscle fibers, and since they have less or fewer of them, it means that it's going to be hard for them to perform that activity. So part D says, explain why calcium ions binding to troponin C allows muscle fibers to contract as in paragraph 5. We know that calcium ions are actually very important in the contraction of muscles because they do bind to troponin C. So here I describe the whole process and how it occurs. I said, when calcium 2 plus ions are bind to troponin C, a conformation change occurs in troponin C and it pulls onto the tropomyosin, and this exposes the myosin binding site. The myosin then binds to actin, forming actomyosin bridges, or you could say forming the actin and myosin cross bridges. These can be called actomyosin bridges. So when this occurs, the ATP on the myosin heads breaks down, allowing for the myosin to pull the actin inward, causing the shortening of the sarcomere and therefore contraction of the muscle. Remember, this is an explanation for why calcium binding to that allows muscle fibers to contract. So it's explaining what happens or talking about what happens after the calcium ions bind on troponin C that leads to the contraction of the muscle. So this information here scores you 3 marks. Let's move on. Here they say, explain how the high energy phosphate provide the energy necessary for muscle activity in paragraph 11. High energy phosphates can include ATP as well as creatine phosphate. We know that ATP, when ATP is converted into ADP, energy is given up, which is going to be used for most of the cellular processes. Also, creatine phosphate is going to be very important because it will provide, like I said here, it provides the phosphate to the ADP in order for it to be converted into ATP for the continuation of muscle contraction. So I said, high energy phosphates like ATP and creatine phosphate are involved in muscle contraction. When ATP is hydrolyzed, energy is released as the phosphate breaks from the ATP, enabling the myosin filament to pull away from the actin molecule. So that is how you get the 2 marks. And next here they say, suggest explanations for the effects that a reduction in tendon stiffness results from aging could have on the movement at a joint. This is about paragraph 12, referring back to paragraph 12. I said, change composition or the makeup of the tendon reduces its ability to recoil at the joint after lengthening. Remember, there used to be lengthening and then the ability to recoil back, or what you call the contractile force. So this contractile force at the joint is going to be reduced, and this softness or less stiffness affects movement at the joint by causing unwanted movements or less movements than what is desired. Let's continue to the next part. The last part says, suggest how myostatin might act as a negative regulator of muscle growth, as referred to paragraph 13. Myostatin is linked to the development of sarcopenia. So here I said, myostatins act as transcription factors that inhibit cell differentiation in muscle growth. Remember, muscle growth is myogenesis. They therefore enhance muscle loss or wasting away of muscle. So remember, if they are inhibiting the formation of muscle, of course, it means they're going to be enhancing muscle loss or wasting away of muscles as proteases break away the muscle protein in there. So if you had written like this or something similar to this, you get the 2 marks here. This brings us to the end of this question 8 as well as the end to this paper. Thank you for being with us. Please do not forget to subscribe. See you in the next video. Bye-bye.